welcome to chapter 2, where we will talk about leaf structure and function. So 2.1 will be will just be about this structure. Very simple, very short. We'll cover the questions and answers towards the end as well. And then 2.2 onwards, it will be main organs needed for the different activities that plants carry out to sustain their survival, I guess, their presence, their being. Okay, so chapter 2. We will explore the structure, as I've said, and then gases exchange, transpiration, photosynthesis, and a bit of an interesting concept, which is compensation point. You can scan the QR code to go a little bit more detailed on what you should know, what you should understand by the end of this chapter. Okay, so a few questions first. How are the external structures of a leaf and internal structures of a leaf lamina? We'll go over that. What is the main organ for gases exchange, transpiration, photosynthesis, and lastly, what is meant by compensation? We will go through this, the explanation to understand with each subtopic of this chapter. Right, so we begin with a bit of a fun fact as usual. So the colors of leaves depend on the pigment molecules contained in them. So pigments are chlorophyll, carotenoid, and anthocyanin. I think you've heard of chlorophyll before, especially with the TikTok trend suddenly booming about drinking chlorophyll for better skin. Why? It's very expensive. Just eat more vegetables, right? Obviously, vegetables contain chlorophyll if they're green. Anyway, the chlorophyll pigment causes the leaf to appear green. So that's the main point of the chlorophyll pigment in leaves, to appear green. So in 4-season countries, leaves are green during summer because light intensity is very high, and during autumn and winter, some plants stop making chlorophyll. The chlorophylls are actually broken down into smaller molecules. So without chlorophyll, right, other pigments, they shine off a bit more, it's their spotlight because chlorophyll is broken down. So your carotenoids and anthocyanin comes into play, causing the leaves to change into yellow and red. So the leaves changing colors, it's about the pigments changing. Okay, the different keywords that you will learn in this video. We will go on with lamina, petiole, the two different mesophils, and then there's also transpiration, microbalance, herbarium, granum, thylakoid, stroma, photolysis, and again, compensation point. Okay, let's start with 2.1, structure of a leaf. So we will only be talking about leaf, okay, one leaf. And then, of course, it's the same for each and every leaf of a plant. So it is the main organ that carries out photosynthesis. Okay, we will be going to in more details in a few subtopics after this. We have your external structure and internal structure. We start off with the external structure. There's only two. You have your lamina, the leaf, as you all know, and the petiole. Petiole is just this small structure that connects the leaf to the main stem or the main body of the plant. Okay, so lamina is the flat, thin, smooth, and green part of the leaf. Of course, the green part is it's green everywhere. Sometimes even the stem is green, especially for herbaceous plants. But yeah, it is flat, thin, smooth, green. It is a flat shape to provide a wide surface, a larger surface area, right, to expose cell containing chloroplast. So chloroplast is the organelles in the cell that contains the chlorophyll that is needed for photosynthesis. So if you have a wide surface area, you expose more chloroplast. You expose more chloroplast to the maximum amount of sunlight, so you have more chlorophyll working for photosynthesis. It's also thin, so it's very thin. I've never seen a thick leaf before. I think. Yeah, they're all thin because it allows gases involved in photosynthesis to diffuse efficiently in the leaf. And then we move on to the petiole, the small thingy here. It's the stalk that connects the lamina to the stem of the leaf, and it stretches out into the lamina, producing a network of middle veins to support the lamina, as you can see here. Right? So it supports the vein itself. The vein of the leaf is not actually the petiole. The petiole sort of stops here. It goes into the vein. It branches out into the veins, but the veins itself is not petiole. Okay, right. So an activity zone, you probably have done this in school, in class, like in your own class. But I feel like it's really interesting if you like leaves, if you like flowers. A herbarium is actually a specimen collection of plants, which is preserved through a certain method. Okay, it's a specific method. It's just dry pressing leaves and flowers between thick pages. A thick book between pages. Okay, you can Google the taxonomy, morphology, ecology, and geography of the plants to make your own herbarium. I feel like if you're living near like a forest or a garden, if you keep a herbarium, it would be really interesting to see if there are any changes between now and the next few years among the species of the plants that are nearby. Just fun activity if you're bored at home nowadays. Okay, so now we go to the internal structure of the leaf lamina. So this is inside the leaf itself, the microscopic structure. You cannot see it with your blind eye. So we start off with the fact that leaves grow without overlapping. Okay, this is to enable that each and every one of the leaf of the lamina receive optimum light to carry out photosynthesis. So such arrangement is of course called leaf mosaic. Okay, mosaic. Of course, there is sometimes a slight overlapping because one leaf is bigger than another, but naturally a plant will try to grow out its leaves without overlapping each other. Okay, it's like a survival structure, I guess, in plants. Okay, so over here we have a nice visual of a internal structure of the plants. You can see the cuticle, you can see the upper epidermis, you can see the layers of mesophyll, the air spaces in between as well. You have your vascular bundle over here, and then the lower epidermis and the guard cells and the stoma that is embedded in the lower epidermis. Okay, so very important thing to remember, epidermis has two types, which is upper and lower, but please remember when it says upper, it's on the side facing out towards the sun, and the lower is the one that is not facing the sun. It is not, for example, this is not the correct one. This is upper epidermis and this is lower epidermis. That is wrong. Okay, so this is upper, this is lower epidermis. Okay, the outermost layer. Well, of course, cuticle lah paling. Cuticle is the most outer layer, and then you go with the epidermis. Okay, epidermis. Right. With that out of the way, we'll go to the explanation of each structure. So, I think we've said something about cuticle before. So it is a waxy, waterproof, and transparent layer. Very important, waxy, waterproof, and transparent layer. It covers the upper and lower parts of the leaf epidermis. Of course, because it is waterproof, it reduces excessive loss of water through evaporation, okay, transpiration, and also it being transparent allows sunlight to pass through it, just to make sure that whatever sunlight passed through it, so that the chloroplast can be exposed to sunlight. Okay, so that's the cuticle. Now we go to the upper epidermis. So this is located, of course, on the upper surface of the leaf, which is under the cuticle layer. This one does not contain chloroplast, and it is transparent, so the light can pass through. It's just sort of another layer of protection for the lamina. Okay, remember, no chloroplast for upper epidermis. So then we have your lower epidermis at the bottom here, located, of course, lower surface of the leaf. And then this part has the stoma and the guard cells. Okay, so the stoma is just a small opening. Okay, and the guard cells is the one guarding the opening and the closing of the stomata. Stoma is one, stomata is many. Stoma is singular, stomata is plural. Okay, I hope that's correct. Yes. And then we will go into the specifics in the next subtopics, okay, of this one, the opening and closing of stomata. Okay, then we have the mesophyll. So first we have the palisade mesophyll. These are arranged vertically and closely packed. Arranged vertically and closely packed to receive maximum light exposure, and they are the sites for photosynthesis. Maximum light exposure, so sites for photosynthesis. So of course they contain many chloroplasts. Then we have your spongy mesophills. So spongy mesophyll is arranged differently. They are irregular shaped, which increases internal surface area for gases exchange. So instead of photosynthesis being the main function, the main activity in that mesophyll, spongy is for gases exchange. So it's a bit irregular shape, loosely packed, okay, loosely arranged, have many intracellular air spaces. So with that characteristic, it can ease carbon dioxide and water absorption through the leaves to the palisade mesophyll cells during photosynthesis, and it does have less number of chloroplasts than palisade. Okay, and lastly we have your vascular bundle, your xylem and phloem, of course. So the walls of the xylem are lignified and thickened to provide mechanical support, and the normal information of what they transport: xylem, water and mineral salts, and phloem, organic substances. Okay, so we do have a few questions here that we can go together. So the first one, name two external structures of a leaf. If I said before, very simple, very short, two answers: lamina and petiole. Okay, what is the importance of cuticles in a leaf? So for cuticle, like I said before, waterproof. It reduces excessive loss of water from the leaf. Okay, there are a few other answers you can put as well in terms of the photosynthesis, about it being transparent, about it being thin, but mostly the main importance would be to reduce the excessive loss. And you can also talk about what you've learned in chapter 1, which is to provide a bit of protection from the leaf. There's no harm in putting that as well. No additional answer. Okay, so number 3, name 5 internal structures of a leaf. So we have your cuticle, and then we have your upper epidermis, your palisade mesophyll, then spongy mesophyll, and then you have your vascular bundle in between as well, and lastly the lower epidermis. So those are actually 6 internal structures that I've said. You can choose any 5. Okay. Number 4, state the functions of xylem and phloem. We have also talked a little bit about that in the previous chapter. Also in the table that we have just covered over here. So xylem transports water and mineral salts, and then phloem transport organic substances. Okay, you can also talk about from where to where. And then number 5 over here, the layer of cuticle and upper epidermis is transparent. Justify. Okay, so why is it transparent, right? It's because we want to make sure that whatever light that the leaf is exposed to, it can penetrate the two transparent layers to reach the chloroplast that is a lot in the palisade mesophyll layer. Okay, so of course it is to allow sunlight to pass through for photosynthesis. It's kind of straightforward, I think, in terms of why it is transparent. And lastly, compare the layers of palisade mesophyll and spongy mesophyll. So for here, you can talk about the arrangement, is it loosely packed? How is it arranged, vertical or random? You can talk about the presence of chloroplast. Is it a lot? Is it a little? And of course, the functions. What is it mainly for? Is it photosynthesis, gaseous exchange? And I think once you fully understand why the layers are arranged in such a way, you can easily connect and you can easily remember the functions of each internal structure, right? Hopefully that was helpful, and that is the end of 2.1. We will go to 2.2 in the next video.